Okay, so since you can't do the Meiosis pop bead lab on your own, I will simulate it for you. So you can watch this simulation and then answer the questions and draw the pictures within the lab as I progress through this simulation. So we're going to start with number 1 on the procedure. I've modified it a little bit. It says create 2 homologous chromosomes using 30 red beads for the paternal chromosomes and 30 yellow beads for the maternal chromosomes. So paternal means from father, maternal means from mother. So if you look, these red ones are paternal chromosomes, these are our maternal chromosomes. So these two would be homologous because they're the same length. So this has 3 beads, 3 beads, 2 beads, 2 beads. This white section is the centromere. And then these two would be homologous, the paternal chromosome with the corresponding maternal chromosome. So you can see same centromere position and same chromosome length. Let me flip that around this way. All right, so total this cell has 4 chromosomes. And remember, if we're in interphase, you technically wouldn't be able to see the chromosomes, but in this simulation, we're going to use them as we can see them, act like we can see them. So normally, if this was G1, there would be chromatin inside the nucleus here. But for simplicity's sake, we're going to look at the chromosomes already. So then number 2 says place your homologous chromosomes on the dry erase board and draw a nuclear envelope, which I have drawn here. Here's your nuclear envelope or membrane. And then draw the cell membrane. So here's your plasma membrane represented by the P in the upper right-hand corner. Number 3, if the cell's cyclin concentration is high enough, the cell will enter S phase. So remember, in Chapter 9 we talked about cyclin and cyclin-dependent kinases. So those regulate or control the cell cycle. And so if there's enough cyclin in the cell, it's going to move on to S phase. So when this moves to S phase, you should recall what happens in S phase. Yes, that's where DNA replicates or chromosomes duplicate. So what we're going to do is we're going to duplicate the chromosome. So there's the duplication there, duplication there, duplication there, and duplication there. So now we have duplicated our DNA in S phase. We have replicated chromosomes now, so we have 4 chromosomes total, 1, 2, 3, 4 in this cell. Each replicated chromosome has 2 sister chromatids. So this has 1 sister chromatid there and a sister chromatid there, and they're exactly the same genetically, meaning they have the exact same DNA sequences. All right, so number 4, once the DNA replication has completed, the cell has prepared for meiosis during G2 phase. It checks to make sure the DNA has been copied correctly. If everything has worked, the cell enters meiosis. So now we're going to move to the first stage of meiosis. All right. So I will simulate the steps of meiosis 1 and meiosis 2, and like I said, you can watch this and answer the questions within the lab as you see fit. If you can't print off the lab, no worries. Just answer all the questions on a separate piece of paper. That's perfectly fine. Right. So, and when we meet in our class scheduled times this week, if you have any questions about this, feel free to ask. I know you're doing this on Thursday, so we'll talk a little bit about Thursday during class, and then Friday, if you have questions, you can ask. So let's begin. So the first phase of meiosis 1 is prophase 1. So that's where homologous chromosomes pair up and form tetrads. So these 2 homologous chromosomes are going to pair up, and these 2 are going to pair up. Make sure the magnets work. There we go. All right, so our homologous chromosomes have paired up. And if you also recall, in prophase 1, you have crossing over that occurs. So these 2 are going to overlap. So we're going to take off the yellow bead there, the red bead there, and they're going to swap genetic information. There's that. so there's the end of prophase 1. So our homologous chromosomes have paired up. We have our paternal chromosome, our maternal chromosome. Crossing over has occurred, so we've created some genetic variation. Also in prophase, the nuclear envelope starts to disappear. So we'll erase that. All right, so now we're in prophase 1. Now the next phase is metaphase 1. So that's where the homologous chromosomes line up in the middle. I'm going to do it vertically here just because it's easier to see. Now remember, another thing that can lead to genetic variation is independent orientation. So if you notice, I have the paternal on the left side here and the paternal on the right side here. It doesn't have to be that way. This could actually have arranged this way, like this. And if it arranges like this, move that over, then that can lead to genetic variation because this side is going to go into different gametes than those chromosomes, as we'll see as we progress through this. All right, so this is independent orientation, along with crossing over, shuffles up the genetic deck of cards. I'm going to put it back how it was, though. All right, so now we're in metaphase 1. So then in anaphase, remember we also have spindle apparatus here, so these spindle fibers are going to attach to the homologous chromosomes. And so in anaphase 1, what happens is we separate the homologous chromosomes. So these homologous chromosomes are going to start to separate and go to the opposite ends. Let me erase these. All right, so remember the whole point of meiosis 1 is to separate homologous chromosomes, which is what we're doing. All right, so and then in telophase 1, they reach the poles. Like so. And then we're going to have cytokinesis occurring too, to where we're going to eventually start to get this look, to where the cytoplasm is also divided. And so now, eventually, after telophase 2 and cytokinesis, what we're going to end up with are 2 cells. Let me erase my spindle apparatus here. Erase this nucleus. All right, so now we have 2 cells, and if you notice, now we have 2 chromosomes in each cell. So what we've done is we've reduced the chromosome number by half. So these are actually haploid cells. So N equals 2. So when we started, we had 4 chromosomes in the cell. It was a diploid cell, 2 sets of chromosomes, maternal and paternal. Now, after meiosis 1, we have 2 haploid cells, only 1 set of chromosomes. And so meiosis 2 is we want to separate the sister chromatids. So this looks a lot like mitosis. All right, so after our nucleus has reappeared, so these are in the nucleus. Remember the chromosomes would decondense into chromatin, and then it would form the chromosomes over again. So now we're going to go into prophase 2. So let me label this. Prophase 2, the chromatin is going to condense back into chromosomes, so we have that here. The nuclear envelope starts to disappear again. So we erase the nuclear membrane or nuclear envelope. It's the same thing. All right. There's no crossing over anything that occurs in prophase 2 because we no longer have homologous chromosomes in the cell. We have separated the homologous chromosomes represented by the different colors. So if you notice, there's not the same length. So here, this homologous chromosome is in this cell, and the other one's in this cell. So in meiosis 1, we separate homologous chromosomes. All right, so now in metaphase 2, metaphase 2, the chromosomes are going to line up in the middle. In anaphase 2, just like in mitosis, we separate the sister chromatids. So we'd have the spindle apparatus again. Like so. And so our sister chromatids get pulled apart in anaphase 2. Like this. Right? And then we move to telophase 2 and cytokinesis. I'm just going to write telophase 2. Telophase 2. Right, so the chromosomes go to the poles. The chromosomes would start to decondense. We're going to get a cleavage furrow occurring. Now we're going to get something like this, where this cytoplasm is starting to separate. And eventually, after telophase 2 and cytokinesis, what we're going to end up with are 2 cells. Let me get rid of all my spindle fibers. Bear with me for a second. So ultimately, we're going to have I have limited space, that's why my cells look more like these ovals. So now what I have are 1, 2, 3, 4 haploid cells, each with 2 chromosomes. 2, 2, 2, 2. And if we look, not one of these 4 cells are genetically the same, represented by the colors, except for none of them are. So if you look, this cell has this paternal chromosome and the larger maternal chromosome, where on the far right we have the larger paternal, larger maternal, or smaller maternal there. And here, due to crossing over, we have differences too, because we've shuffled up the deck a little bit. And so not one of these chromosomes or cells is genetically identical. And so the whole point of meiosis is basically you're producing, through 2 cell divisions, 4 genetically different haploid cells that we call gametes or sex cells. So if this was a male, these 4 cells would be sperm cells. If it was a female, one of them would become an egg cell. Right? So that's the process of meiosis. And so what you would need to do is you can pause this video to help you draw the pictures that you're going to have to do. It says you need to sketch them. You don't have to sketch each individual bead, but I would recommend, if you have the ability, to use the colors that are associated to. So find a yellow marker and red marker, or yellow colored pencil and red colored pencil, and follow the same color pattern that was simulated by the Bead Lab when you draw these pictures. All right? And as always, if you have any questions or if you're unsure of something, do not hesitate to ask me. Thanks for watching. Have a good day.